It's a pleasure
Hi.
to be here with you all this evening and opportunity to talk a little bit about might be something you would be interested in adding into your health program. So we're going to show a few slides here, go through those as we talk about it. This is probably the least important one of those that we'll talk about today. So we'll just skip right ahead. So transfer factors. Let's go back to the beginning. And this isn't something that For Life discovered. This is actually something that was a discovery of this gentleman, H. Sherwood Lawrence, a doctor at New York University. He had been spending most of his career to this point looking at infectious disease and looking for ways to kind of help people stay healthy, to kind of protect them, and looking for ways to help prevent illness in individuals. And so in 19, well, actually, it was about 1944, 45, he started looking at white blood cells. Now, these are the immune cells that circulate in your bloodstream. So all day, every day, these are going around circulating throughout your body and are one of the main ways that immune cells move through the body and one of the ways that they carry immune messages from one location to another. And so we thought this is probably a good place to start. We'll look at the immune cells within the blood. They're easy to collect. We can concentrate them fairly quickly, and we'll see what happens. And so he started off his initial research looking at a disease called tuberculosis. Now, at that time in the US, it was one of the major infectious diseases and was responsible for quite a few number of deaths each year. And so this was a good target to start off with. And he noticed that there are some people that contract this illness and they recover and do fine. And he wondered, was there something perhaps in these white blood cells that could be given or taken from someone who survived and given to someone who had never been exposed and thereby pass that protection between individuals? So his first couple of studies were simply that. He took someone who had recovered from tuberculosis and he pulled out their white blood cells, he concentrated them, and then he injected the whole cells back into a naive person, someone who had never been exposed to tuberculosis. And like many of you, he gave the little tiny injection under the skin a few days later and then waited to see if it turned red, a standard tuberculosis test. And sure enough, within a couple days of giving those white blood cells, it did show a positive immune response. And so he had proven his initial theory that there was something in those white blood cells that could be transferred between people. Well, he did a number of other researches past that, trying to better understand. And then in 1949, he took those same white blood cells and he broke them open and took the contents of those cells and then ran them through some really crude membranes, basically the ones they were using at the time for dialysis, people that were diabetic, and thereby concentrating some proteins from within all the contents of those immune cells. And when he got down to the certain fraction of proteins, he discovered that this is where all of that activity lied. So all those other fractions didn't seem to have that same transfer of information that this one fraction did. And he coined the term transfer factors. He had found these factors in white blood cells that could transfer immunity. And so that's where the name comes from. That's where these molecules were first discovered. And it spawned a ton of research for probably the next 30 to almost 40 years. There was a ton of research done on it. Some claim as many as 3,000 studies. So far, we've been able to track down about 1,800, almost 2,000 of these. I've been able to add those into our collection as a reference on the research that's been done on transfer factors. And fast forward to today, now you've got over 70 years of research on these molecules and all the benefits they have. This counts to millions, probably hundreds of millions of dollars worth of research on these small little proteins that come from our white blood cells, the immune cells in our bodies. So let's talk a little bit about what it was that Dr. Lawrence identified. So back and forth for the first probably 20 years of that research, they were trying to decide exactly what this was. Was it a piece of DNA? Was it a bit of the RNA? Was it some other kind of cellular signaling molecule? And the research kind of settled in on these are likely very small proteins. And they sit somewhere in the 3.5 to 12 kilodalton size. Now, kilodalton is kind of a little bit of a confusing term. Don't worry about that. There's a slide coming up where we'll talk about what that really means. But this is a way of measuring the size of molecules, and it's a very, very small measure. And so, as just as a kind of comparison, the antibodies that we've all heard a lot about over the last few years are about 10 times this size. So you're talking over 100,000 daltons in size for an antibody. And here we're talking about something that's around 10,000 daltons. So about a tenfold smaller molecule than the stuff that's been in the news left and right for the last several years. So I kind of put the picture of the flowers up here, as I think this is a good analogy to kind of help you understand what transfer factors are. Now, it's not a single molecule. Transfer factors are actually thousands, maybe hundreds of thousands of similar yet different molecules. And so the flower, I think, fits well because there's something almost every flower has in common, and that's the stem. Right? There's the flower on top and this stem that goes down. And regardless of what flower it is, what plant it comes from, the stem looks very similar from one plant to the next. It's pretty straight, has a few leaves on it. That's pretty standard. And transfer factors are a lot that way as well. So there is what they call a conserved region to them. So there's this part of the molecule that is consistent from one transfer factor to the next. But then you have that flower on top. And depending on what plant it is, what time of year it is, all that things, you have a wide variety in what that flower could look like. The length of the petals, the color, the shapes. Maybe it's more than one color. It's a small flower, it's a big flower, all that kind of stuff. And transfer factors have that same variability. So you have the conserved region, that constant part, and then there's another piece of the molecule that is highly variable. And this is the specific part of transfer factors. This is the part that carries specific immune information to your immune cells. And really, this is what makes transfer factors unique and very different from everything else out there, in that they're providing specific information to your immune cells. Most of the other things you'll see in the marketplace, and we'll talk about this a little bit more later on, but they're there to kind of just give a general push to kind of lift up the immune system a little bit, to activate it, maybe rev things up a little bit faster. But it's not really improving the efficiency with which the immune system works. It's really just kind of either giving it more muscle, giving it more speed. Transfer factors are providing education. Right? So that highly variable thing or portion of the molecule is specific to a particular item. Right? So for an example, a virus has a number of unique features to it. We've heard a lot about spike proteins in the past couple of years. That's one example. But you have things in the genetic material within the virus, other things on the surface of that virus particle, anything that your body can identify as being unique to that particular invader, it can create a transfer factor toward. And that variable region is going to be kind of like a key and lock. So it's fit perfectly around whatever it is, so that when it comes in contact with it, there's that perfect fit. And it then helps those immune cells make a quick identification as to those things that can be a threat to it. The immune system actually spends quite a bit of time making sure that it has correctly identified friend or foe. And you want it that way because you don't want your immune system just going off and attacking anything and everything that it comes in contact with. You want to make sure that you're attacking those things that really do pose you a risk. And so there's a lot of energy and a lot of safety checks that your immune system has to make sure that it makes that correct identification. Transfer factors are one piece of that. And by having those from the outside, you're essentially teaching your immune cells this is a known threat. Therefore, when the immune system comes in contact with it, it already knows this is bad, it needs to go. You don't have to wait the usual couple days to sometimes a couple weeks for the immune system to make that identification and begin the immune response. And so that's one of the factors of transfer factors. We'll talk about this again a little bit later. So we talked about the Daltons. Now let's kind of give you a little bit of a better, hopefully, picture of what that really means. So we're going to start big and work our way down small here so you can see just how small a Dalton is. So here's your average 60-kilogram woman. That's something we're all fairly familiar with. Now, if we go 60 times smaller, down to the size of a melon, weighs about 1 kilogram. So we've gone 60-fold smaller. Now let's go down 1,000 times smaller into something, a paperclip. It weighs roughly 1 gram. Now this is again something we're all pretty familiar with and have a good idea of what 1 gram feels like. And these are all measurements that I think most everybody are familiar with as well. So nothing new here. Now we're going to get into something that maybe you're not that familiar with. For those of you that have had an experience have experienced snow, maybe you've had a little snowflake on your finger. Well, again, depending on the size of it, but it's roughly 1 milligram. So now we've gone down another 1,000 times smaller. And for those of you that haven't had the opportunity to experience snow, think about a single grain of rice. Now, if you take that little grain of rice and you chop it up 30 times, so you have 30 pieces, one of those pieces is 1 milligram. So we're talking pretty small stuff here. But that's not far enough yet. So next time we go down into the size of transfer factors, we're going to tack on another, 10 or so different zeros to the side of it here. So now, if you're keeping track at home, we're down to 600 quadrillion times smaller than you or I. So that's a huge number, and it's very hard to comprehend just how big that is, or in this case, just how small transfer factors are. So to put that another way, that essentially means that you have trillions of transfer factor molecules in any capsule of our transfer factor products. So there's plenty of these molecules to go around in each of the capsules, plenty to go, and then educate all of those immune cells throughout your body. So hopefully that gives you a little bit of a picture of just how small transfer factors are, how many you can pack into an individual capsule, and the potential that has for educating and enhancing or just generally supporting the health of your immune system. So let's talk a little bit about that education. So again, comparing to some of the other things in the marketplace that have a direct stimulation or otherwise are just non-specifically altering the way your immune system works, transfer factors are giving a specific information to it against a specific threat. Now, it's still up to your immune cells to decide what to do with that information. And you have hundreds of different types of immune cells, each with their own specific role and function in the overall immune response. And it's that coordinated response then that keeps you healthy and deals with anything that occasionally makes it through your defenses. So this kind of has two different effects on your immune cells. One, it can have a stimulating effect like some of those other things, right? So once your immune cells have identified the threat, then they can begin the response. They're going to step up their activity and move forward towards getting rid of that invader. The other thing is it can have a redirection, right? So your immune cells sometimes, for lack of a better description, might get kind of bored. And so they're looking for things to do. And maybe something happened and it misidentifies a potential threat and starts attacking something that really isn't a threat to you, isn't an enemy. So by giving this education, then you can take that focus away from the thing that isn't a threat and move it towards something that really is. And so that's kind of a redirecting of how your immune system responds to things, again, keeping it focused on the things that really do pose a threat to your health. Now, the education that transfer factors provide lasts as long as your immune cells do. Now, different immune cells have different lifespans. They can go from just a few days to some cells may actually live as long as you do. There's still some discovery left in that side of things, but for sake of argument here, we can say that they last for several years, maybe a couple decades. And it all depends on the type of immune cell and the function that it has. But if we kind of average that all out, in roughly about 90 days, you're replacing most of your immune cells. So about every 3 months, you have more or less a new immune system. And so what that means is that every single day, old immune cells are dying and new immune cells are being born. It all takes place in the bone marrow, where all of your blood cells are produced. And these cells then need to be educated. And your body has some methods in there to do that, to train them in different things, and mostly to make sure that they can make that correct identification and they don't begin attacking your own cells. But transfer factors then can help supplement that role or that action of your immune system by then educating these new cells against those potential threats. Again, the end result is that you have a better educated immune system and something that's able to more quickly identify those things that could potentially be a threat to your health. So let's talk about for life transfer factors and how we get them. Now, the original research, as we talked about, took transfer factors from blood. That's an expensive, and there's lots of risks associated with doing that. So it's not really an economically viable way to do it and provide it in large scale like we do. And so there were some researchers that had been doing the standard blood-based transfer factor studies, and in 1989, they filed a patent for getting transfer factors from cow colostrum. And so they had identified transfer factors as being present in colostrum like they were in those white blood cells from the 1940s in Dr. Lawrence's work. This is the patent then that David Lisenby came across a few years later and was the beginning of For Life. And so with a proprietary process, we're able to concentrate the transfer factors out of colostrum. And you can see kind of a little animation here that shows how that happens. It's just filters. There's no chemicals involved. There's no modification of the molecules. We just take the colostrum from the cows. It's pasteurized for safety, and then it's run through a series of ever smaller filters to concentrate down those very small proteins. Now, in the process, it also concentrates a few other things. Anything that is really small in colostrum will also be present in this mix. One of those things that you may be interested in is lactose. So that is also present here. It's just naturally occurring in colostrum and is small enough that it also passes through those filters. Now, the kind of side note to all of that is we're talking some pretty small amounts here. Now, remember we talked about the snowflake weighing a milligram. In each individual capsule, there's maybe 30 to 50 milligrams. It's a pretty small amount. And for most people, that's not going to be a concern, especially if you're taking these with food. But there is going to be some there. It's just naturally present in the colostrum, and it does pass through the filters. But again, it's at small enough concentrations that for most people, it's not going to be a concern. The other place where we get our transfer factors is from egg yolks. So this was actually something that David Lisenby had theorized early on in For Life, is that, well, if we can get transfer factors from colostrum, can we not also get them from eggs? And so with a little investigation, we determined that they were concentrated in the yolk. This is, of course, the part of the egg that feeds the growing chick and gives it all of its nutrition. And it stands to reason then that it would also provide immune information for that developing chick. Now, one thing to note, there's a difference between cows and chickens. I'm sure you're all very well aware of that to begin with. But cows have the opportunity to nurse their young for a number of months after they're born. Now, while colostrum is really high in transfer factors, it's also present to a lower extent in milk. But this means that the cow then can continue to educate the calf's immune system for several months after birth. And so we like to say that in order to give the calf the best chance, the cow has a wide variety or a breadth of immune information that it passes on to the calf. So it wants to cover as many bases as possible to make sure that the calf has as much knowledge as it possibly could need to survive. Chicken, on the other hand, once that chick hatches, it's entirely on its own. Its mother cannot provide any additional information. And so in that sense, it provides kind of a depth of immune information, essentially giving as much as it can on the things that pose the greatest risk to that chick, so that hopefully if it comes in contact with something that it's most likely going to, it will have enough knowledge about it to protect itself. And so obviously if you take that breadth and the depth and you mix them together, you get a very good combination. And that's what we do. And we'll talk about that a little bit more on the next slide. The last thing here is the nanofactor, which is kind of the newest addition in the transfer factor line for life. And nanofactors are a bit different. So they're about, well, half the size of transfer factors. So we're going even smaller with these. Now, they don't seem to provide the specific information that transfer factors do. Instead, based on the information we have so far, they tend to work more on the overall coordination of your immune response. Now, I like to compare this to the difference between a high school band and a professional orchestra. You give them the same pieces of music, the same instruments, and ask them to play it. Are they going to sound the same? No, they're not going to. Right? The symphony has much more practice. They know how to use the instruments well, and they know how to play them together. They're all on the same beat. They're all playing the same note at the same time. It's all very well coordinated. The band is still working on fine-tuning those aspects of it, and that's one of the major differences to it. And so the same thing goes with your immune system. If it is well coordinated, then you get a very efficient, a very effective response. If some things are not happening as fast as they should or at the right times, things don't start or stop at the right point, then you don't get as an effective an immune response. And so nanofactor, from what we've seen so far, seems to aid in that coordination. So the overall immune response is working better together, each cell performing its role when and how it should. All right, so let's talk a little bit about the TriFactor Blend. That's what happens when you mix all three of these together. And based on the research we've done, this shows some synergies between it. Again, with that breadth and the depth, by putting that combination together, you seem to get a more effective mix of transfer factors. And then with the nanofactor there to aid with that coordination, it just seems to have some additional benefits in overall response. This is a patented blend as well. And so before we move in and talk about products a little bit, let's kind of wrap up what we've talked about with transfer factors. And these are really the points that make transfer factors unique, make them different from everything else out in the marketplace. The first of these is the fact that it is made by the immune system for the immune system. So we're taking transfer factors from colostrum, ingesting them, and then they're being distributed throughout the body. But your immune cells are making transfer factors every single day. Anytime they come in contact with something that they don't recognize, they're going to start making transfer factors towards those unique characteristics of whatever that might be. And that is one of the ways that immune cells talk to one another and how that infection information is passed throughout the body. And so there's nothing else out there in the marketplace that can say that. This is something that's coming from your immune cells, and we're just adding to it, giving your immune cells additional knowledge that they may not already have. We talked about the specific information side of it. Again, something unique to transfer factors. It's not making your immune system stronger. It's not pushing it faster. It's educating the immune cells, helping them better understand what it is they face and helping them be able to make that quicker recognition. So we talked about the stimulation and the redirection. So this is keeping your immune system focused on those things that really are a threat to your health, and hopefully keeping it doing its job in the most efficient way possible. We didn't really talk about this in specific detail, but it really is kind of nature's first food, right? Colostrum is something that mammals produce the first 1 to maybe 3 days after birth. And so if it's safe enough for a newborn infant to consume, it should be fine for the rest of us that might be a day or two older than that. And so there is a kind of a built-in safety with transfer factors in the fact that this is something that babies get within minutes to hours of birth. And then we kind of like to wrap everything up with our 3 R's, which are recognize, respond, and remember. We've talked a lot about recognize so far, and the fact that transfer factors are there to help the immune system make that recognition faster so that the immune response then can take hold quicker. And it really does turn out to be a numbers game. So the more time the invader has to duplicate and spread throughout the body, the harder it is for your immune system then to deal with it. So if you can recognize it quickly and you can begin that response quickly, you can limit the numbers of the invader that need to be destroyed. And then the last part of this is the remember. You'll notice, and here kind of the good example is chickenpox. For most people, it's something we usually get as a child. You go through it once a week, maybe two, and then you usually don't ever get it again. And it's not because you don't get exposed to it again, but it's one of those things that your immune system has a very long memory for. And every time you get exposed to it, it kind of reminds your immune system, oh yeah, there's that. But it begins that response immediately, produces the antibodies and other compounds to deal with the virus so that you don't ever get the disease a second time because your immune system has remembered it from that first time. And transfer factors aid in that process as well, because again, it's kind of a reminder to those immune cells. Oh yeah, I remember that thing. And so it keeps the memory of the immune cells going so that when you do come in contact with it, again, your body can start that response quicker, and hopefully you don't have any symptoms of it because your immune cells took care of it before it became a problem. And so there you go. There's a quick little rundown on transfer factors, where we get them, kind of what they do. There's a lot more we didn't talk about today, but hopefully that gives you a good idea of what makes them different, what makes them unique, and why transfer factors are something you might want to include in your health program. So now let's kind of dive in and talk a little bit about products and the research that we do behind them. So we'll start off with on how 4Life deals with the science that we put forth in our products. And over the last probably decade or so, we've been putting a bigger emphasis on research. And what you see here is a list of many of the collaborators that we work with in our studies. This is not a complete list. It grows every single year as we add new labs, schools, and other research institutions to the mix here as we further study our products and further learn about transfer factors. And I put this slide in just to kind of show it's not just 4Life that's doing this research, but we are utilizing the instrumentation and the expertise of many other researchers and university folks and specialized labs to help us in this respect. And it also then adds a bit of a third party, I won't say endorsement, but support for what we're doing, in that it's not just the scientists here at 4Life that are doing it, but there are others outside. And as we've done this, we have pushed these folks to publish the work that we have done. And you can find many of these publications. There is a section on the US website that lists all of the publications we have to date. It is constantly changing. Right now, we're kind of averaging probably 2 to 3 studies every year on our products. And some of the ones just this past year, I think we have 2 that are due to come out in publication within the next couple of months, are kind of methodology ones, as we have tried to better understand transfer factors. you can look at the research we've done in that respect and utilizing in those studies. I think we've had 3 or 4 collaborators help us in doing that. But this just gives you a quick little idea of what some of those publications look like, including the PDR there in the middle. That's the Physician's Desk Reference. It's a publication here in the US that most doctors have in their office and is their first resource when wanting to know what a new drug does and how they should prescribe it and use it. There is a section in the back of this book that is for dietary supplements, and for life transfer factor is the first entry in that section. So if your doctors have access to this, it's a great resource. They can get a really quick, it's about a 1.5-page monograph on transfer factors, summarizing a lot of the things we've already talked about, and then giving the citations to the research behind all of that. So a great way for them to become familiar with what transfer factors are, how they work, and where they can learn more about them. And then all of these publications that you see, they're published in what are called peer-reviewed open access journals. So let's talk about those two things. Peer-reviewed means that we write it up, we either do the research ourselves, work with other companies in collaboration, write the whole thing up, and then it goes out to 2 or 3 experts in the field. They read through it, look at how we did everything, look at the conclusions we've stated, and then they say, all right, we agree, you've done this well, this is valid research. Or they say, what about this? Or you need to say something about this. How did you handle this particular concern? And there might be some back and forth with us on that to explain to them how we did it, adding an additional paragraph here or there so that that information shows up in the manuscript. And then sometimes we even have to go back and do a little bit more research to address their concerns and then add that piece back into it. So typically the peer review process takes between 2 and 6 months, depending on how many questions they have and how much back and forth there is. But in the end, you get a study that is well done and has the backing of not just the scientists who did it, but again, 2 or 3 external experts that agree that it was done well. And the second point there is the open access. This is relatively new in the publishing industry. It's maybe 2 decades old at the very beginning, but becoming more and more popular now. And what this is that means that For Life pays all of the copyright and publication fees up front. And then that makes it free for everyone else to read. So you don't have to pay to read these articles. You can just follow the links, go straight there and read them for free. And so in the past, it was all the publishers paid all that. And so if you wanted to read the article, you had to pay them $20. And nowadays it goes up to as much as $50 and $60 per article to read. And so we cover those costs up front so that you can use it as you will. You can pass that along to your doctors and other scientists if they really want to know how we did it and what the study actually says.